In this video, we're going to talk more about light and pigments and their role in photosynthesis. The previous video you watched introduced you to photosynthesis as a process that converts light energy into chemical energy. Now we're going to take a closer look at how photosynthetic pigments capture light energy. Light is a type of electromagnetic radiation. This is the electromagnetic spectrum here, and what it shows us is the range of wavelengths of electromagnetic radiation. Visible light is the portion of this electromagnetic spectrum that humans can see. Now, light acts as a wave and can act as a particle. So here we see visible light described as ranging from shorter wavelengths to longer wavelengths. So let's sketch some waves so we can see what we mean by wavelengths. So imagine waves at the ocean, right? They have a peak, they have this bottom portion here, a trough. You can measure the distance between the peaks, and that's your wavelength. I also mentioned that light is particle-like, and a photon is a particle of light. Each photon and wavelength has a specific amount of energy. Let's look at this in our visible light spectrum here. So shorter wavelengths have a higher amount of energy, and longer wavelengths have a lower amount of energy. Now, when light hits an object, the photons can be absorbed, transmitted, or reflected. So here we're looking at sunlight hitting a chloroplast, and notice that the thylakoids are reflecting green light, which means it's bouncing off, and transmitting light, which allows it to go through. They are absorbing other wavelengths of light. Let's look at this in more detail. So photosynthetic pigments absorb light, and what we are looking at here is the absorption spectrum. So what we are plotting is the wavelength of light here on the x-axis, and we are measuring the action spectrum of photosynthesis by measuring how much oxygen is produced. So this light tan color is showing you essentially the rate of photosynthesis across wavelengths of light. So notice, right, we have a higher rate of photosynthesis in this violet to blue range, as well as the red light range. Our graph is also showing you specific absorption spectrums for chlorophylls. There are two of them, chlorophyll A and B, as well as an accessory pigment, carotenoid. Notice chlorophyll A and B together absorb in this blue to violet range, and they also absorb the red light. So these pigments are the two that are primarily responsible for photosynthesis. So where do we find these photosynthetic pigments in a chloroplast? Photosystems are the functional units that capture light energy, and they contain pigments. Let's talk about where we find them. So here's a model of a chloroplast. We've cut out the outer and inner membranes, and now we're looking at the inside of the chloroplast. Remember, it contains these stacks of thylakoids, and if we look at just one thylakoid and cut through the membrane, that's where you'll find photosystems. So photosystems have three main components: antenna pigments, a reaction center chlorophyll, and some associated proteins. The role of these associated proteins is to keep all of these pigments and the reaction center chlorophyll gathered together. So most of these photosystems contain 200 to 300 antenna pigments. Antenna pigments are light-gathering pigments, and they guide light energy towards this reaction center chlorophyll. And the role of this reaction center chlorophyll is to absorb that energy and pass it on to an electron carrier. And so this arrow is referring to the fact that it's passing its energy on to an electron carrier, and we'll talk more about that in class. So to summarize, light is a type of electromagnetic radiation, and light can behave in a wave-like and particle-like manner. Pigment molecules are responsible for capturing energy. Each pigment absorbs photons of particular wavelengths, and the chlorophylls are the primary pigments involved in photosynthesis. You find these pigments in photosystems. They're composed of accessory proteins and antenna pigments whose role is to transfer absorbed light energy to the reaction center, where an excited electron is transferred to an electron acceptor.